Today we are going to properly start learning about the genetics. Now, first thing which we are going to learn, that is how crossing over produces variation. Now, if you guys remember last time I told you guys, that let's suppose if you are 3 brothers and sisters or 4 brothers and sisters, all of you are not exactly the same. Even though you have same parents, same mother, same father, why we are different from each other, right? Answer is very, very simple, because of crossing over. Every time when the meiosis takes place, crossing over happens. And because of the crossing over, every time different types of sperms will be made and different type of egg will be made. How we are going to discuss that? Let's suppose that this is a homologous pair of chromosomes. What is this? This is a homologous pair of chromosomes, and it is during the S phase, right? During the S phase of the meiosis, it happens this. We have learned that already, right? But during the prophase 1, crossing over takes place, right? And synapses and bivalent is formed. Do you remember that? Yes. All those concepts which I gave you. Now let's suppose that here, this has gene E, this has gene A. This is also capital E, this is capital A. Now this one over here has small e, small a, small e, small a. Means that the alleles over here, they are dominant, and alleles over here, they are what? Recessive. Alright? Now what happens? That these chromosomes, they overlap their chromatids. Do you remember this or not? Now what will happen? That this overlapping will take place, right? Now, when the overlapping will take place, remember that this was capital E, this was capital A. This was capital E, and this chromatid is going over here. This is capital A. Understand? This is small e, and this over here is small a. This is small e, and look here, this chromatid is coming over here. This is small a. Now what happens? When this chromosome is pulled in this direction, this chromosome is pulled in this direction. So these chromatids, they will become separate from each other. And at the end we get what? 4 separate cells, right? Now in all those 4 separate cells, there will be 4 types of gametes. Now, there will be 4 combinations. In one combination, this capital E and capital A, this one, look here, capital E, capital A. This chromatid will be completely over here. No change in this one. This one, capital E, it will get exchanged with what? Small a. This small a. So, it will become what? Capital E, small a. Then this small e, he will get what? This capital A. So he will become what? Small e, capital A. But this one, small e, small a, it will remain the same. So this is small e, small a. So if we observe, what has happened? 2 gametes, they are exactly like parental. They are exactly like parental. But 2 new combinations have been formed. 2 new combinations have been formed, right? Now, as these 2 new combinations have been formed, if this gametes forms, you see, joins, let's suppose this is a sperm, joins with the egg cell, so what will happen? A different kind of zygote will be formed, right? Which will create or generate a variation. Now, let me tell you one thing. Here in this diagram, I showed you guys crossing over at one point only. But crossing over can take place at more than 1 point. Like, let's suppose that this is another pair of chromosomes, right? And look here, has happened? At two places crossing over is taking place. At two places crossing over is taking place. Another way I can show you, let's suppose that this is another pair of chromosomes. So there is what? Crossing over here as well, here as well. Understand? Not only the lower chromatid is getting crossed over, upper and lower both are being crossed over. So crossing over can take place in one pair of chromosomes at more than one places. So it means that there are so many chances of what? Of producing new types of gametes. So when there are so many chances of producing new types of gametes, then what will happen? More and more chances of variations are there. Now, question is variation good or bad? It is good, right? Because variation, we will learn that in the next chapter as well, inshallah, in evolution and variation. That because of variation, new characters they come in the organism, right? And new characters, they may help, again I am saying the word, may help in survival from the changing environment. From the changing environment. Now, the weather is changing continuously, right? It is becoming more and more warmer, right? So, what is happening? So, those organisms which will be able to cope with the hot weather, they will be best fit, right, in that particular environment. That is only possible if variation is taking place. If those organisms, there is no variation, simple mitosis is taking place or no crossing over is taking place, what will happen? No change in the organism's body, no variation. So eventually the person will become unfit for the changing environment and that species can vanish from the face of the earth. So variation is good. Now, so everyone understood that how crossing over causes the variation. That what happens? Shuffling of genes takes place. Shuffling of alleles takes place because of crossing over. Yes.
So crossing over will take place between alleles of the same type?
Yes, exactly. Always. Because the locus is what?
Same.
Permanent. Locus is same for all the genes. Understood? So, and remember one thing, that when they will be crossing over, they will be crossing over from the same loci. Understood? So this happens for all these. Now remember that here I just gave you about the two alleles, right? Let's suppose that there was over here what? Another allele called capital G. Over here it was small g. Here it was capital G, capital G. Let's suppose that, right? Now what will happen? Now when this is capital G, this is small g, this is capital G, this is capital G. Now on this one what will be there? This will be small g on this one, rest will be what? Capital G, capital G, capital G. Now is it just like parental? No. Now what has happened? Difference has come in the gametes. So in this example you are just looking at 3 genes. On one chromosome there can be thousands of genes. They will be shuffled, right? So when they will be shuffled, huge variations can take place. Understood everyone? Now, next we are going to discuss 2 things. Now we are going to start learning about, first of all, Mendelian genetics. Now what is Mendelian genetics? Now Mendel, he was actually a teacher and a priest. He was, you see, a son of a farmer, and in his days, the old concept was there. You see, when the son will grow up, the son, he will help his father in the fields, will not study, right, and do whatever the chores the person have to do. But with the help of his sisters, he convinced his father that he want to study. So, you see, his father, he sent him to Austria. In the Austria, at that time, there was a monastery. The people, you see, right now, as we have madrasas in our Pakistan, right, in India, in subcontinent. So before, and even you see before, like 500, 600, 800 years ago, the education was given in madrasas, right? If you hear the word of Al-Khwarizmi, right? He is a Muslim scholar, right? Muslim scientist. And other, you see, scientists, they were studying in madrasas. But they were not only studying Quran and Hadith, they were also studying about science as well, right? So just like that, the Christians, they had monasteries. In their monasteries, they used to get the knowledge of their religion, but they also used to study science as well. So what happened that, you see, this Mendel, he went to Austria, he took admission into, you see, monastery, where he started studying about science. And when he finished his qualification, he became a teacher over there. But he was not a good teacher. Remember, you see, everyone cannot become a teacher. A person who gets a PhD degree, right, he has immense knowledge, but if he do not have the art to deliver that knowledge, he is a good, you see, scientist. That person may have immense knowledge, but if he do not have the capacity to deliver it to the students, he is not a good teacher, right? It does not mean that the person is bad or something. Same, you see, the Mendel, he was not a good teacher. He could not deliver in the class, but he was a good scientist. He was a good observer. So what did he do? In his spare time, he started planting tea plants in the garden of the monastery. Now, we have madrasa, they had monasteries, right? Where the students, they will live, they will learn their religious education and scientific education. Now, if you guys remember, yesterday I told you that he was lucky that he studied how many traits? 7, right? If he would have studied an 8th trait, his all experiments would have gone wrong and he would have rejected his own ideas at that time. And he may have not published them, right? So, few of the traits that he studied, one of the traits was height of the pea plants. Let me mention over here, height. Another trait that he studied, that was shape of seed. Another trait that he studied, that was color of seed. Another trait that he studied, that was color of flower. Now, 1, 2, 3, 4, there were total 7 of the traits, right? 7 characteristics that he studied. And luckily, there are 7 homologous pairs of chromosomes are present in the pea plant. There are 7 pairs of chromosomes in the pea plant, 14 total chromosomes. Now, after his whole study, now you see, for being a scientist, not only you have to be a good observer, you have to have a good mathematical mind as well. Now, in biology as well, we will study a little bit of statistics. Now, why statistics? So that we can plot the data. No? You know that you learn how to draw a graph. You learn how to make a table and record the readings, right? That is actually a part of mathematics, that is called statistics. So you have to have that knowledge as well. And he was good at that as well, in keeping the record, noting down the data as well, and then concluding the results from the data as well. So he was lucky that he studied 7 traits and there were 7 pairs of chromosomes. Now, after all his studies, he gave 2 theories which later on became laws as well. But now we know that one of those is not a law. One is called as law of segregation, and second one is called as law of independent assortment. First of all, we are going to talk about law of segregation. Who will tell me? Now this word segregation, it is derived from an English word segregate. What is the meaning of segregate?
To divide.
Separate. Right? To divide or to separate. Now, he said that when reproduction takes place, at the time of reproduction, there are factors that control the traits. Now, at that time, there was no concept of gene or allele. This is 1860s. He started his work at the end of 1840s, right? And he finished his work and he published his work in 1860s, right? So, what did he say? That at time of reproduction, the factors, now at that time the genes and the alleles, they were called as what? Factors. He says that at the time of reproduction, factors, they separate from each other. We know that now. How? Look here. E is an allele. It becomes separate from the other allele. Does it become or not? Understand? So he said that at the time of reproduction, factors which we now call as what? Alleles, separate from each other. Right? What is law of independent assortment? Now, the law of independent assortment tells, now if you guys remember, yesterday I told you monohybrid cross and dihybrid cross. What was monohybrid cross? If you study one trait at one time, that is called monohybrid. Understand? And if you study two traits at one time, that is called what? Dihybrid. Understood? Now this law of independent assortment he gave on the basis of his dihybrid studies. In the beginning, he studied only one trait at a time. Right? Now when you are playing a video game, first stage is easy, right? The second stage becomes what? Difficult. So when he became obsessed with the first stage and he finished the first stage of the video game, when he conquered the monohybrid studies, so let's go and make it a bit difficult. Study two characters at the same time. But this is the time when he was lucky. Now, what is the law of independent assortment? Says that the genes or alleles of two traits assort independently from each other. Now what does this mean? Complicated words, right? Not complicated. According to this statement, he says that each gene is present only one chromosome. One chromosome has one particular character. And remember that at that time there was no concept of chromosomes as well. Because chromosomes were not discovered at that time. The microscopes were not that much powerful to discover the chromosomes at that time. Right? So he just said that he did not have any idea. Even he did not have idea about the genes and alleles, right? So he used the word what? Factors. So he said that at that time. Now I am writing this definition according to modern times. He would not write the genes or alleles, but he would have written factors. He says that the two traits, their factors, they always assort independently. They do not affect each other. They do not affect each other. The height do not affect the shape of the seed. The height do not affect the color of the seed. Or color of the seed and shape of seed, they do not affect each other. This is what he tried to say. We are going to prove it with the help of experiments, right? Now, first thing, let's talk about law of segregation. We can take the example from the plants as well. We can take the example from the animals as well. First, let's take the example which the Mendel used. What did he do? He took tall and short plants. Now, remember yesterday, I told you guys that he took what? The true or pure breeds. Now, what is a true or pure breed? Who remembers? Very good. A homozygous organism for a particular trait is called true breed or pure breed. Now, what did he do? That he crossed a tall plant with a short plant. In result, he got all the tall plants. Now, when in result he got all the tall plants, sorry, what he came to know, that the tall trait is what? Dominant. He came to know, because you see, in F1 generation, all the offsprings, they were tall. I am calling it as F1 generation. Now, a new question comes in your mind, sir. What do you mean by F1? First, filial generation. The full meanings are first filial generation. Now, a new question comes in your mind, sir. What is the meaning of filial? The word filial, it means offsprings. First generation of offsprings is called what? F1 generation. What is it called? F1 generation. Now, whenever you get all tall, it means tall is what? A dominant character. Now, from here we can move forward. We know that tall was what? Homozygous. We can write that its genotype is capital T, capital T. And the short genotype, it is what? Small t, small t. By the experiment, we came to know that short is what? Recessive. And according to the definition of recessive, which I taught you yesterday, a recessive can only express itself in homozygous condition. A recessive can only express itself in homozygous condition. So it is small t, it expresses itself, right? Now, this is what? This is the phenotype of these organisms. This is the genotype. Next step, in the genetic diagram, we always write what? Gametes. Now, in the gametes, what happens? Alleles separate from each other. So what will show? Capital T circle, capital T circle. Small t circle, small t circle. Now remember one thing. You have learned this in all levels, but you guys did not appear in exams. So you don't know, you don't have the idea whether you can lose the marks or gain the marks. If you do not circle the gametes, -1 mark. If you do not circle the gametes, -1 marks. So remember that you have to circle the gametes. Next, what do? We make either a Punnett square. Now this is called as a Punnett square. Now here we take male, here we take female. This is the symbol for the male, this is the symbol for the female, right? Now sometimes when you go to the malls, outside the public washrooms, they have made these symbols, right? So this symbol represents male, this symbol represents the female. Now, let's suppose that over here we have capital T and capital T. Here we have small t and small t. Right? Now, this capital T joined with this small t, it becomes capital T small t. We know that capital T is a dominant. So this one will be what? A tall plant. This capital T and this small t will join capital T small t. Now remember one thing. Whenever you are writing a genotype, you will write the dominant allele first and recessive allele afterwards. These are the rules that you have to follow. So this is capital T, small t. Again, this will be what? Tall. Again, this capital T, small t, tall. Capital T, small t, tall. So F1 generation, all of them are what? Tall. But they are all what? Heterozygous. They are all what? Heterozygous. One allele is dominant and other allele is what? Recessive. Now, what do we do always? After getting the progeny or the offsprings from the F1 generation, we always cross F1 with F1. Now, here it means that we are crossing the tall plant with the tall plant, right? So remember that this is what? This is the phenotype. Next step, what we are going to do? We are going to write the genotype. What is the genotype of this tall? Capital T small t. Capital T small t. Understand? Now, over here, next step what we are going to do, we are going to write about the gametes. So here the gametes will be what? Capital T small t. Capital T small t. Now, here I taught you about the Punnett scale. Now I am going to teach you about the another method by which we can solve this problem, that is the cross. That is called cross. Now, how we can do that? Please pay attention. This capital T joined with this capital T. So it will be what? Capital T, it will be what? A homozygous tall plant. Understand? Then this capital T will join with this small t and it will become what? Capital T, small t, and this will also be what? A tall plant. Now first one has joined with these two, right? Its job is done. Now the second gamete, small t, will join with this capital T, it will become what? Capital T, small t, it will be what? Tall. And then this small t joined with this small t, it becomes small t, it will be what? Short. So this over here method is called Punnett scale method. This is called cross method. Understood? Now we can see from here and we can get the tall ratio short. There are 3 tall plants and there is 1 short plant. Now let me tell you interesting fact. At that time when he performed these experiments on the plants, pea plants, the plants he got, he recorded their numbers and he had exactly the same ratio. Exactly the same ratio. He have written this in his you see findings. Exactly 3:1. Now, when he had these tall and short plants, 3:1, if you see that one of these plants, one out of these tall plants, one of these plants is what? Homozygous. And rest of these 2 plants are what? They are tall but they are heterozygous. Now let's suppose that there are 2 tall plants in front of you. Rather, let me make 3 for you, right? 1, 2, 3. This is tall, this is tall, this is tall. By looking at this tall plant, can you tell which one is homozygous, which one is heterozygous? From the phenotype, you cannot tell the genotype. From the phenotype, you cannot tell the genotype. But as a scientist, you have an intriguing nature. You want to find out whether the plant I am looking at is homozygous or heterozygous. Question is, how will we find it out whether the organism under observation is homozygous or heterozygous? Yes. No.
With a short plant.
Yes.
You cross it over.
What is this called? Well, very good. This is called as test cross. Now, there is a definition of test cross. There is a definition of test cross which I am going to explain it to you guys. Right? First, let me write down the definition, then I will explain it to you. The definition is cross between the organism under observation with its recessive parental type is called as test cross. Now, organism under observation is what? A tall plant. When we cross it with recessive parental type, now what is the recessive parental type? Short plant. Understand? That cross will be called as a test cross. Now guys, I am going to teach you about the test cross, and we are going to make two rules out of this. Right? We are going to make two rules out of this. Now I am going to rub this area. Alright guys, now there are two possibilities. There are two possibilities. One possibility is that the tall plant can be capital T, or tall plant it can be capital T, small t, right? A tall plant can be either homozygous or heterozygous. So we are going to take first possibility. What is the first possibility? That the tall plant, which is actually what? Organism under observation. Let's suppose, now what are we doing? We are supposing that this is what? Capital T, capital T. Suppose, right? At the end we will get the rule out of it. Now, let's suppose that this is what? I am writing organism under observation. Instead of writing organism under observation, I am writing OUO. Right? Which is what? A tall plant. Understand? We have to cross it with what? Recessive parental type. Now, what is the recessive parental type? That is a short plant. Now, if what happens, we cross this tall with this short, which is the organism under observation, and what is this? We have supposed that this is what? Capital T, capital T. We already know that the short is what? Small t, small t. We already know that the short is what? Small t, small t. Now, what will happen? Now, this is what? This is genotype. Next, what we will write? Gamete. So, gamete will be capital T circle, small t circle, small t circle. Now, we have already made a cross for this over here, right? But very quickly for you guys, I am making a cross over here, saving some space. So what do we have over here? Capital T, small t, all of them they will become what? They all will be tall. Now please, this is the important point to understand now. we had an organism under observation. We did not have any idea whether it is homozygous or heterozygous. We crossed it with what? Short plant, and all the plants in this test cross we got what they were? Tall. They were tall. And we get tall only when we have a true breed. We get only tall in a cross with the short, only tall when the tall is also what? A true breed, a homozygous organism. So from this point we can prove one thing, that organism under observation is what? Homozygous. So from here we got one rule. If all the offsprings of a test cross are dominant. Now here you see we are making a generalized rule right now, not only related to this. So I am writing dominant, in bracket I can write tall, right? For this trait, if all the offsprings of a test cross are dominant, then organism under observation is homozygous. Now you will understand it more when we will perform the second possibility. You will understand it more when we will perform the second possibility. Now let's come to second possibility, then I will come back to this, right? Now the second possibility. Now again, what do we have? We have a tall plant, right? And let's suppose this is what? This plant is heterozygous. We are supposing this right now. Now, this tall plant is organism under observation. We have to cross it with what? Recessive parent, right? So, this is tall plant, right? And this is short. The genotype we have supposed what? Capital T, small t. And short genotype we already know. It will always be what? Small t, small t. Now, when the gametes will be formed, the gamete over here will be what? Capital T, here it will be small t. Here it will be small t, small t. Now, I am going to make a Punnett scale on a side. Now, this is interesting over here. Capital T, small t. Capital T, small t. Now, please pay attention. This capital T and capital T, they will join together to make capital T, what it will be?
Sir, it's small t equals to small t.
Oh, sorry. Small t, small t. So, it will be capital T, small t. It will be what? A tall plant. This small t and small t, they will join together to make what? A short plant. This capital T and this small t, they will join together to make what? A tall plant. This small t and small t join together to make what? A short plant. Now, we take the ratio of tall ratio short. How many tall are there? 2. How many short are there? 2. We simplify and we get ratio 1, ratio 1. It shows that 50% of the organism of this test cross are dominant and 50% are recessive. Understand? So we can make a rule. Second rule from here. Now this is the rule number 1. Now we are going to make the rule number 2. Now the rule is, if in result of a test cross 50% of offsprings are dominant and 50% of offsprings are recessive, then organism under observation will be heterozygous. Now, forget this cross, forget this cross. Forget this cross, forget this cross, which we have performed for our understanding. Let's suppose I had a tall plant. I crossed it with the short plant. All the plants I got in result, they were all tall. Simply I can tell on the basis of this rule, the plant that I had, the tall one, is homozygous. Is it what? Homozygous. Next, I had another tall plant. I crossed it with the short plant. The new progeny that I got, the new offsprings that I got, 50% were tall, 50% were short. According to this rule which we have devised, the organism that I was observing is what? Heterozygous. You won. You became a scientist, geneticist. Right? So, we can find this out. So, we have to remember these two rules for our social life. Understand? But genetics is not that much simple. When the epistasis will come, that will be a different story. But we are talking about simple genetics. We are talking about simple genetics, right? We can again, you see, I told you that this is the example we are taking from the plants. Now, I am giving you guys an activity to do right now. Let's suppose that there is a grey dog, right? And there is a white dog. Now, if we cross them, in the F1 generation, they all are grey dogs. If we cross them, in the F1 generation, all are what? Grey dogs. And remember that these both are what? Pure
So grey
breeds.
dominant.
So what does it mean? It means that grey is what? Dominant. Now I want you guys to perform the F1 test, F2 generation, right? And then do these both, this possibility of, first possibility and second possibility of this test cross right now.
F2 generation test means F2 cross with
Yes. No. F1 with F1. You see, produce F2 progeny, F2 generation.
what is filial? Yeah.
The filial means offspring, right? Progeny. Progeny means when we get offsprings in huge quantity. We call it as progeny.